Hello, APBIO. This is a tutorial just to patch up a little part that we didn't get to in W4, and anybody else can listen to it too. This is a conversation about our free response question that I gave you. I want to make sure you understand part B. So we got through all the different parts. We talked about part A in class, we talked about part C in class, but I want to address part B. So in part B, the question asks asks you to compare prokaryotic cells and eukaryotic cells. It says that I'll read the question. Prokaryotic cells lack membrane-bound organelles found in eukaryotes. However, prokaryotes must perform many of the same functions as eukaryotes. For three of the organelles identified in part A, explain how prokaryotic cells carry out the associated functions. So when we talked about A, hopefully now you would be pretty clear as to which organelles should be present in eukaryotic organisms. You should be able to come up with pretty quickly the nucleus and mitochondria should both be there. And then if you were looking ahead to B, you might also say ribosomes. That would make B easier to answer. For the fourth one, for A, there's all sorts of things you could choose. But for B, if we were to think about the difference or comparing eukaryotes to prokaryotes, you can't choose some of the membrane-bound organelles their functions in prokaryotic cells are outside the scope of this class. But for the nucleus, the mitochondria, those are going to be definitely tell you that they're eukaryotic cells. So that's a really good answer to A. And then ribosomes are in all cells. So that's another logical example or a specific choice for A. So let's talk about those three if you were to choose those for B. First of all, say you chosen the nucleus. So in the nucleus, now your job in B is to compare. It specifically says they have to perform the same functions as eukaryotes, and then you have to explain how prokaryotes carry out the same associated functions. So you actually have to define the function and compare the functionality, and then talk a little bit about how the anatomy is different, but how it accomplishes the same thing. So in the nucleus, the function of the nucleus in eukaryotes, here we're going to compare the two. If you were to choose that one, it contains the genome. So it has the DNA in it, and the DNA is the blueprint for proteins. And that's the same thing. In prokaryotes, there is an organelle that contains the genome, and it is a blueprint for proteins, but that's the nucleoid. So you can see here that I've said that it's the same function. It contains the genome and DNA, and it's the blueprint for proteins. But instead of a nucleus like we have in eukaryotes, we have a nucleoid in prokaryotes, and you can see a diagram of that over here. You notice in this diagram, this is really out of scale. This is 0.1 to 10 micrometers, and this is 10 to 100. So I don't really like that, but it does show you this nucleoid region as compared to the nucleus. Now, the big difference in structure is that this nucleus is membrane bound, and in the prokaryotes, it's not membrane bound. So the question is, how does it accomplish the same function? How does it have all this genetic material in it? And, why doesn't it quote unquote need to have a membrane around it? Well, if you look at the genetic material of eukaryotic cells, there's multiple linear chromosomes. And in prokaryotic cells, there's a single circular chromosome. So it's so much, you can't argue that it's hard to keep track of one thing. It doesn't need a bag around it, essentially, because it's just one thing, one circular chromosome. And because these are smaller and simpler, they don't have as much DNA. And so there's simply less DNA. It's only one piece of DNA, and it's circular. And so the fact that there's not a membrane around the nucleus doesn't inhibit its function, which is to contain the genome, the DNA, and be the blueprint for proteins. Now, the other thing that's interesting to note, and would be a more comprehensive answer, is you could talk about the simplicity of prokaryotic cells. They don't have the capacity to specialize because they have less genetic material, and then they only form unicellular organisms. Eukaryotes have the capacity to specialize, and they form unicellular or multicellular organisms. So that's another difference between the two. These guys are just more simple. They have less genetic material, and because of the anatomy of that genetic material, they just don't need a bag around there. They function fine without having a bag around their genetic material, essentially. Okay, So now let's look at the next thing. If you were to choose mitochondria, that all eukaryotic cells have mitochondria. So if you saw mitochondria, you would know it would be diagnostic that it was a eukaryotic cell. So that would be an excellent choice for A. In B, if you were going to talk about how prokaryotes do it, you'd have to say what the function of the mitochondria are, which is it's the site of cellular respiration. It produces ATP. Now, if we were to look at prokaryotes, all organisms require ATP. All cells. Pardon me. Well, we could say organisms. All cells. All cell processes run on the same type of cellular energy, which is ATP. So prokaryotes need to make ATP too. And the question is, how do they do it? So instead of having this mitochondria, they have something called infoldings. And then some of the reactions happen right in the cytosol. Because truthfully, some of the reactions happen in the cytosol for the eukaryotes too. But if we were to look at those infoldings, these are some electron micrographs of prokaryotic cells. And they just have infoldings, which are part of their plasma membrane. So this is showing you a cross-section of an aerobic prokaryote. Not all prokaryotic cells have infoldings, but they just use their plasma membrane, folds on their plasma membrane as the surface area and as sort of makes these pseudo compartments to generate concentration gradients for the process of ATP production. So you can see here how that happens also in photosynthetic prokaryotes, because some prokaryotes can carry out photosynthesis, and they don't have chloroplasts because they don't have membrane-bound organelles. But over here we have aerobic prokaryotes. So these guys are going to use oxygen to make ATP, just like eukaryotes do, but they don't have a mitochondria. Instead, they have these infoldings, and then some of the reactions happen right in the cytosol. Now, the question is, well, how do they do it without, how can they make enough ATP? And the answer is, again, they're unicellular and they're smaller, so they don't have as much energy requirements. The eukaryotic cells are larger, many are multicellular, and they have larger energy requirements, so they need to have a more, they need to be able to produce more ATP, and they can because they have the mitochondria, and the prokaryotes can't. So it does limit the size, and it limits the ability for prokaryotes to specialize, and then they can't make multicellular organisms. Okay. And then the last thing is ribosomes would be a third excellent choice for B. We know all cells have ribosomes because all cells need to make polypeptides or proteins. And this is the first step. In eukaryotic cells, ribosomes synthesize polypeptides. In prokaryotic cells, they also synthesize polypeptides, but they're slightly different. If you look over here at this diagram right here, it's showing you that the S unit is the unit that's used to measure the size of ribosomes. It's just a weird unit. You don't have to know that, but you should see that 70 is a smaller number than 80. So the prokaryotic ribosome is slightly smaller than eukaryotic ribosomes. Here we have that the larger, these are smaller. That doesn't really have any implications except that it does the exact same thing. It's just a slightly different structure. And also in eukaryotic cells, we have both free and bound ribosomes. The free ribosomes are making proteins that stay right and are used right in the cell. The bound ribosomes are making proteins that are either inserted into membranes or secreted from the cell. There's a lot of cell specialization because there are multicellular organisms, and so those cells, those specialized cells, have to be able to secrete proteins in eukaryotic organisms. However, in prokaryotic organisms, most of them are free. There can be some bound to the plasma membrane, and that's how they get proteins in their membranes. That's how the proteins are inserted into the plasma membrane in prokaryotic cells. But for the majority are free, and that's because the proteins are made and deposited right in the cytosol. There's less proteins made because there's less energy required, and there's less process because they're more simple. And they don't specialize, so there's not a lot of secretion of proteins that are required, and most of the proteins are just produced for cellular use. Here you can see here how there can be ribosomes this is showing it bound to the plasma membrane in prokaryotic cells. But again, this doesn't doesn't happen as much in prokaryotic cells. It's just not necessary because they're so much more simple and don't form multicellular organisms. Okay, that's it for B. I hope that's helpful.